Good afternoon and welcome to the Spotlight on AT Research webinar. It's organised by Action for AT. My name's Naga Monchetti, broadcaster, journalist and charity patron, and I'm joined here in this studio by the Chair of Action for AT Research Advisory Committee, Dr. Mark Toms. Hello.
Hi Naga.
What should we expect to get out of this session? Everyone who's joined us, and thank you everyone who has, but what should we expect?
Well, really what we're hoping to achieve is to connect the AT community around the globe and at the same time really try and get the opportunity to showcase some of the great research that's going on.
Okay, and I know we've got some great speakers coming up as well to do that, and hopefully some good questions as well from all of you. Mark and I are going to be with you for the next couple of hours. Let's show you the agenda, shall we, for today's event. You'll see it up on your screen in a moment. Here we go. We have a full programme, a great lineup of speakers from all around the globe. They're going to showcase their current studies in a concise and easy to understand manner. That was important for me. That was the rider I put down, as well as answering all of your questions. And all the presentations are going to be shared afterwards. And of course, you're going to have a chance to ask questions after each presentation. We've got Slido in there, as well as we're going to get you to vote on various polls too throughout the event. So if you haven't used Slido before and you're a little bit confused, here's an easy guide to it.
We're excited that you're joining us for this digital event today, and we really want you to get involved. There are a number of ways you can do this. Use the round Ask button in the bottom right-hand corner of the page to post a question. You can do this with your real name or post anonymously. If you want to upvote a question that's already been asked, use the thumbs up button. Throughout the event, our speakers may activate polls which will appear automatically. Please do get involved with these so that our speakers know what you're thinking. We really hope you enjoy this event today, and now it's time to introduce our first speaker.
So I'm delighted to say we have speakers from all around the world, from the UK, the USA, Israel, Spain, Australia, and Mark and I would like to know where you're tuning in from. So let's see your whereabouts across the world you're viewing in this webinar. And we've done that clever thing with that word cloud. You've seen these go around. So if you can click on the polls tab on the top right hand of your screen, if you manage to do that, and answer the question, there'll be other polls, of course, throughout the event, and we can find out where you're all coming from. I think the main place you're all coming from for the moment, here in the UK and in Spain as well, South Africa, Norway. Gosh, Mark, did you expect so many people from all over?
It's fantastic to have so many people from around the world.
So much interest as well. So look, thank you, welcome, and do stay with us for the next couple of hours. It's great really to have so many people from the community of the AT representatives. We're going to begin the formal presentations in a moment, but here is a short message of welcome and thanks from the Chairman of Action for AT, Toby Reid.
Hello and welcome to the Spotlight on AT webinar. My name is Toby Reid and I am one of the two founders of Action for AT, the medical research charity that has funded the research we're going to be talking about today. Firstly, I want to say thank you to Naga Monchetti, Dr. Mark Toms, who will be hosting today's webinar, and also to the researchers you're going to be hearing from. Thank you as always for the time you give to our cause. So Action for AT was set up in 2012 by my wife Emily and myself in response to what so many of you will be very familiar with, the devastating news of a diagnosis of AT for our then 5-year-old daughter Evie. So in the time that we received the news, we decided what we wanted to do was fund research into trying to establish whether there was a cure or a treatment. And we felt we needed to do that because we weren't able to identify a huge amount of research going on in this space. So we took it upon ourselves to set the charity up, and with a huge amount of support and goodwill, we've managed to now fund over £3 million worth of medical research projects, 40 in all across the globe. And you're going to hear about 4 of those today from some of our experts that we've managed to be able to support. So I'm absolutely delighted that we can hear exactly and directly from them today, and look forward to what I hope will be a really, really useful and informative session for everyone. Thank you.
We all do. Thank you very much, Toby. It's now time to learn a bit more about the current AT research studies which are taking place at various institutions around the world. It's time for you to sit back, enjoy. Please don't forget to use the Q&A button on your screen to ask any questions. Up first, we have Dr. Rob Dineen, who is an NHS doctor, consultant radiologist and Professor of Neuroradiology, try saying that quickly, at the University of Nottingham and the Queen's Medical Centre in Nottingham. Dr. Dineen.
Good afternoon, everyone. And firstly, thank you to Action for AT for inviting me to talk about our research. My name is Rob Dineen, and I'm a doctor and researcher in Nottingham. My job involves using MRI scanners to find out why people are poor and to help guide the treatments that people receive. Many of you may have come to Nottingham to visit the National Children's Ataxia Telangiectasia Clinic, where we have a team of experts who can help the child or young person with AT and their family with all the medical aspects of AT. It's a great pleasure for me to be able to work with this excellent team. Some of you may have had an MRI scan before, and so will know what's involved. For an MRI scan, you can lie down on a couch and go into a tunnel. These pictures show our new MRI scanner, which looks pretty space age. You can see the couch you lie on and the tunnel that the bed slides into. You don't have to do anything apart from lie still while the scanner takes pictures. It can take a while to do the scan, often half an hour or more, and can be a bit noisy, but while you're having a scan, you can watch a film. If you look at the picture on the right, what film do you think is playing? That's not a member of staff at the back of the scanner. That's a screen behind the scanner showing the film Shrek. And if there was a person lying on the bed, they'd be able to watch the film using a special mirror that lets you see the screen. MRI scanners are pretty amazing. As well as taking really detailed pictures of the inside of your body, they also allow us to make lots of different types of measurement that tell us how your body's functioning. In our catnap studies, which some of you may have taken part in, we've used MRI to understand changes in the brain in people with AT. MRI scanners work by using powerful magnets. To show the powerful magnets in action, for a bit of fun, we put a wire wool pan scrubber inside a tennis ball. Watch how even this small amount of metal in the tennis ball gets pulled by the magnet. It looks like the ball is stuck to my hand, but in fact I'm stopping it flying in. When I tilt my hand, off it goes. But the fact that MRI scanners use magnets and not X-rays, like CT or PET scanners, is really important for people with AT, because people with AT have a higher sensitivity to X-rays. Because of this, we want to use X-rays as little as possible, and if you like, to save X-rays for when we really need to use them. Because MRI is very safe for people with AT, it's the ideal type of scan for us to use for research and clinical care for people with AT. As you know, AT is a condition that has a number of medical aspects. This is because the ATM protein, which is the key protein in AT, has important roles all over the body and in maintaining the health of different organs. We know that there are changes in the brain and lungs leading to difficulties with movement and breathing, and in Nottingham, we've used MRI to study both of these. The immune system can be affected, leading to an increased risk of infections. But for our current study, we're looking at a different one, which is that people with AT have a higher chance of developing certain types of cancer. A previous study has estimated a risk of cancer of up to 23% before the age of 20 years in people with AT. This means that around 1 in 4 to 1 in 5 people with AT will have a diagnosis of cancer before the age of 20 years. Although cancers in childhood are very rare, we know that there's a basic principle that the earlier a cancer is detected, the earlier treatments can be started, and as a result, the outcomes are improved. In spite of the increased chances of developing cancer in AT, there is no agreed national or international guidance for screening for cancer in people with AT. So what do we do at the moment? Well, in many centres, including at the Nottingham Clinic, blood tests are performed that can detect some types of cancer. This is certainly an important thing to do. One problem is that the chemicals in the blood that can tell doctors that a cancer may be present could be elevated in people with AT for other reasons. In addition, there are other blood tests that could detect cancers which are not currently part of the blood tests used. In Nottingham, we're developing a different approach. We think that doing a whole body MRI scan could allow us to detect cancers before they have caused noticeable problems. The idea is a bit like the screening programs for breast cancer or cervical cancer in adults that you may already be aware of. whole body MRI scans like these can be taken in around 20 to 30 minutes. The pictures on the left are pictures that we've taken of a healthy volunteer using our new scanner. Hopefully you can see the outline of the person, and all the darker areas that you can see are normal healthy organs. However, if you look at the scan on the right, this is a scan of a young man, and you can see some dark blobs that shouldn't be there. These dark areas are due to cancer that has grown within the nodes in the chest. This person doesn't have AT, but they do have a type of cancer that can occur in AT called lymphoma. Next slide, please. So how are we going to go about doing this? We think that a screening program using whole body MRI scanning and blood tests could be a really good way to find cancers early in people with AT. But before we can set this up as a service, we need to do a series of studies to show that this approach is feasible and that it works. Over the next year, we will be looking at the feasibility. As shown in the purple box in the middle of the screen, we are going to be inviting 20 children and young people with AT to come to Nottingham to try out the scan. We really want to understand what the experience of having the scan is like and whether there is anything we can do to improve the experience. We'll also look carefully at the images to see how well the scan has worked. Because this type of scan hasn't been used widely in AT before, we don't know how sensitive it will be for picking up the things that shouldn't be there. If you are already coming to Nottingham to visit the AT clinic, then that is the perfect time to come along for the scan, but we can also arrange visits at other times. We also have a really nice children's waiting area, and for people who are a bit nervous, we have a pretend MRI scanner that you can practice on so that you can get comfortable with going into the tunnel. That's shown bottom right of the screen. But before we start doing any scans, there's a really important piece of work we need to do first. We want to understand how people with AT and their families would feel about the idea of a screening program for earlier detection of cancer. We realise that different people affected by AT may feel differently about what finding a cancer would mean to them and how it would impact on their lives and the lives of their loved ones. For this reason, we will soon be inviting people to share their views with us, either in a one-to-one chat or in small groups. Understanding the concerns and views about a cancer screening program is vital to help us to design how to do this further down the line. And if it all looks positive, we'll move to the next stage, which will be a larger and probably international trial of this screening approach, looking at the outcomes. Such large trials are usually needed before clinical guidelines can be changed. One really important question that we need to consider is this a crazy idea? Well, I don't think so, because this approach has been used before in another condition. Li-Fraumeni syndrome is a genetic syndrome that also has an increased risk of cancer during childhood. Li-Fraumeni is different from AT because it doesn't affect the brain, lungs, and immune system in the same way. The types of cancer that occur are also a bit different. But when the trials were carried out looking at using MRI for cancer screening, they found that those taking part in the MRI screening had a much better outcome than those who didn't. It is now in the international guidance for people with Li-Fraumeni to have the screening scan, and we hope that our studies will show a similar benefit for people with AT. Next slide, please. So please do contact us at the email below if you want to share your views on cancer screening in AT. And please also get in touch if you are a young person with AT or you have a child with AT and would be willing to come to Nottingham to have the whole body MRI scan. When you email us, we will send you more information about what is involved, so even if you are not sure about taking part at the moment, you can take your time to think about it and get back to us if you want to join the study. We will also be linking up with international AT experts to find out what they're doing about cancer detection in their own centres. We want to draw on their expertise to help us answer important questions about how a screening programme should work. For example, how old should a child be before the first scan? And how often should the scans be performed? And we really want to know whether they would be interested in joining us for a larger trial in the future. Finally, I should point out that all of this work has been made possible by the amazing support from Action for AT, and in particular, the David Peake Appeal. I'd also like to acknowledge my fantastic collaborators here in Nottingham, and to introduce Renata Neves. She's the one who is the PhD student working on this project, and a fully qualified NHS MRI operator, and she's the person be taking all of the amazing MRI scan pictures. Thank you very much for listening.
Rob, thank you so much. I'm allowed to call you Rob, aren't I?
Absolutely, yeah.
Fabulous. So Mark and I have established our double act already, right? So I get to ask the really simplistic, dumb questions, and Mark's going to ask all the smart questions. So that's fine.
That's
so
a deal.
Rob, here's my first. It may seem very obvious to you. You said that, you know, when you're looking out for people in the trials to take part and feedback, you're going to look at how often children would be scanned. In your ideal scenario, take all the money out of it, all the funding out of it, how often would you like to scan those with AT in terms of cancer detection?
I mean, that's a really good question. It's a very important question. And I think the honest answer is we don't know, because as far as I know, nobody's tried this before. I would say my gut feeling is that, you know, we may end up scanning, for example, once a year. That would seem to me to be about the right amount. But that's just based on my own knowledge, my own experience and gut feeling. And I think what we really want to do with engaging with the international expert community is to find out what they think and see whether people think that's reasonable. And then if there's agreement with that, or 2 years or 6 months, or, you know, we would then design a proper trial based around that gap between scans to see whether that works or whether we need to adjust it. I think we're at the very early stages here. There's still a lot of things that we don't know and we want to find out.
And in terms of practicalities, in an ideal world, would that coincide with, say, annual checks?
Ideally, I think just to reduce the number of visits that families have to come to the hospitals, it would be nice if they, you know, coincided with visits to the hospital. Of course, one of the things about this type of scan is that it can be done at pretty much any hospital that has an MRI scanner, a modern MRI scanner, and so people wouldn't need to come to Nottingham to have the scan. Once the program's up and running, they would hopefully be able to go to their local hospital, just to reduce the amount of travelling people have to do.
Mark.
Thanks, Rob. That's really helpful. Now, you may have covered this a little, but you mentioned in your presentation that you'll be looking at the feasibility of whole body MRI scanning as a screening program. Can you just explain to us a little bit more about what you mean by feasibility?
Yeah, absolutely. So whenever you're conducting a clinical trial, before you undertake a very large, very time-consuming and potentially very expensive trial, you want to know that the thing that you're testing in your trial basically does the job. You want to know that it can get nice, clear images. You want to know that it can show you the sorts of things that you're looking for, in this case, cancer. And of course, we want to know that the test is acceptable to the people who are having the test. So for the children and young people who are having the scan, we want to know that it's something that is going to be well tolerated, that people aren't going to find unpleasant. So the feasibility trial is just a small scale trial. In this case, we're looking at recruiting 20 or so children and young people to really answer those questions, to find out how people find the experience, so that our experts looking at the scan can check that they're getting all of the information they need from the scan. And if it all looks good, at that point, I think we get the green light to move on to a much larger study that will hopefully give us the definitive, the final answers on whether this is the right thing to do.
Rob, we've had a question come through on Slido, and of course we've got so many people from all around the world listening today, and understandably, Isabel, thanks very much for engaging, is asking, do you know if there's this type of MRI scanner in Spain? Yeah, so the MRI scanner that we're going to be doing this particular test on is just a standard MRI scanner, which most hospitals would have. This particular type of scan is probably best done on a newer scanner rather than an older scanner. So, but you know, most hospitals that have MRI scanners should be able to perform a scan like this. So, I think it's perfectly feasible that it would could be performed in most hospitals in Spain that have MRI scanners.
Thank you, Rob. And we have another question, and obviously our audience are aware that radiation is something that can be a problem in patients with AT. So the question is, if the MRI uses magnets, does that mean that there is zero risk of radiation to the patient?
Yeah, the good news is it does. There is absolutely no radiation, the X-ray type used in MRI scanners at all. I mean, I do need to be a bit careful here. There are some risks with MRI scanners, and so before anyone goes into an MRI scan, they always have to complete a safety questionnaire, mainly to make sure they don't have any metal on their body or within their body that could cause problems, because obviously metal and magnets do potentially interact. But no, there's absolutely no X-ray radiation in at all. So from that point of view, they are very safe.
So that's reassuring. One more question. We are getting lots through, and we'll try to get through as many as we can. Will you share the results of the study publicly so they can be used by other professionals from other countries?
Absolutely, yes. That's really important for us. So, we always try and publish our scientific research so as many people as possible can see what we found. We do hope that if we go on to conduct a much bigger trial to test out this approach, that other centres, people in other countries will join our study and work with us. But if people want to conduct their own studies, then of course that I think would also be brilliant.
Thanks so much. Dr. Rob Dineen, thank you very much for your time and for answering those questions with grace and simplicity, may I add, my gratitude for that. So that's Rob there. Thank you very much as well for sending in your questions. Keep them coming in, because this is all about what you're interested in as well. We're going to move on to our second presentation. Please welcome, live from the United States, the Principal Investigator of the Global AT Family Data Platform, Executive Director of the US-based AT charity, the AT Children's Project, Jennifer Thornton. Jennifer, welcome. Thank you very much. What time is it where you are?
Oh, it is about 11:30 in the morning.
Okay, so quite a decent time. Thank you for joining us. Please take it away.
It is. Thank you so much. I appreciate it. As she said, I'm Principal Investigator of the Global AT Family Data Platform, and also I've been the Executive Director of the AT Children's Project for over 20 years now. And it's been my very great honor and privilege to work with so many of the AT families. We started the Global AT Family Data Platform a few years ago when the AT Children's Project founder, Brad Margus, wanted a way for families to be able to share their information about AT directly with researchers. By giving the power to the families to share their data, we would be able to build a platform that would give researchers access quickly with as little red tape as possible. Our objective was for researchers to use this data to make discoveries about AT, and we're hoping that those discoveries could lead to treatments for AT and that we would be able to move quickly. Next slide, please. Now, because AT is such a rare disease, we're encouraging families from all over the world to participate, because it's really only by joining forces that we'll have enough data to help researchers discover treatments. So we're hoping to continue to recruit as many families as possible, and that's one of the reasons that I really appreciate the opportunity to come and talk with everyone today. We've tried to make it a relatively simple process for families to participate, and this is a picture of the landing page. This is what you'll see when you go to atfamilies.org. Next slide. If you're an AT family and you'd like to get registered, the first step would be to create an account. Next slide. Next slide. Then the website will walk you through giving permission or consenting to participating on the platform. Next slide, please. Then you'll be asked to answer some questions to give a brief overview of your child's medical history. Then we'll get your shipping information so you can participate in the whole genome sequencing part of the platform. All right, next slide. The genomic sequencing is taking place at the Broad Institute at MIT and Harvard in Cambridge, Massachusetts in the United States. After the person with AT provides the saliva sample, the collection kit arrives at the Broad Institute. Sorry, go back one slide. Thank you. The collection kit would arrive at the Broad Institute with just a code assigned to it. They don't have any names on them, so you can be assured that it would come without any identifying information. Then after the DNA is sequenced, the information ends up on a secure cloud, also with just a code assigned to each sequence. All right, next slide. Thank you. Now, what do we mean by whole genome sequencing? What we mean is that we sequence the entire genome, not just the ATM or AT gene, but everything. And why is that? Well, there are a couple of reasons. So one is that ATM is a very long and complicated gene. I think most of you probably know that. But there are over 600 known mutations within the AT gene that are known to be disease-causing. So we can learn which patients have which ATM mutations, which is really important. And then we can also identify those hard to find mutations. Some of you may know that it's not always easy to find AT mutations. So I think of it as almost like it looks in the nooks and crannies to really make sure that we can identify the hard to find mutations as well. Also, we're hoping that we can identify modifier genes. That means genes other than the AT gene that can impact AT. Find out why some people get certain symptoms and others don't. And this information will hopefully give us some clues as to how to treat AT. Next slide. So in addition to the sequencing data, we have plans to add other types of data to the platform, and we're really excited about those as well. As a matter of fact, we are actually launching our first quick survey very soon. Came back from the translator a couple of days ago, so we're hoping to have that ready to go in a few weeks. Next slide. So in our effort to enroll as many patients as possible from around the world, we're translating the platform into many languages, and it's actually available in 12 languages so far. Next slide. And so far, we've identified patients in 43 different countries who are all participating. It's always wonderful to check email in the morning and find a new participant enrolled, especially when it's from a country where we previously hadn't been in touch with any patients or doctors, which happens on occasion. And if you'd like to help us recruit families in your country, we would really love that. If you could, you'd reach out to me and we could strategize on how to best do that. That would be really wonderful. Next slide. So anyone diagnosed with AT from any country can register and enter their medical history on the platform. However, for the whole genome sequencing part of the platform, we currently have approval to include people with AT from a handful of countries so far, which is 11 different countries, and we are working on more. So please do be patient with us. We're working on it. If you live in a country where we do not yet have an approval for the sequencing part of the project, please do keep in mind that when you log onto the platform in the medical survey, there is a blank field where you can enter your child's AT mutations if they are known. Also, you can email us a copy of your child's mutation report if you have it, and we can share it with researchers as possible. It doesn't replace the whole genome sequencing, but it's very helpful to have in the meantime. Next slide. This is from a couple of years ago when I had the chance to go to England and attend their family weekend. I recruited dozens of people with AT in person for the platform, which was a lot of fun. Here are two of the participants with their saliva collection kits. They provided samples. We shipped them back to the US for whole genome sequencing. And it was really wonderful to meet so many families in person and explain the importance of the project. Next slide. Looking forward to the day when we can meet again in person. So what does it mean that this is a patient-driven effort that's overseen by families and experts? Well, it means that no one particular doctor or hospital controls the data and decides which researchers can access it. It's families who put the data in, and it's families who decide to share their data. AT parents from 10 different countries sit on the Family Oversight Board, and these advisors are the ones who appoint our experts, the members of the Scientific and Medical Advisory Board who review all of the data access requests. Next slide. And researchers are currently accessing the data, so the vision has come to life. If any researchers are watching, if you have a good idea and would like to access the data, whether it's about the various ATM mutations or modifier genes or clinical data, for whatever reason, you could go to atfamilies.org, click on the For Researchers drop-down menu, and submit a simple application. It's a relatively quick process. We've been known to get researchers access sometimes within 24 hours, so no promises, but it is a relatively quick process. Next slide. Just want to say thank you to our donors and our grassroots fundraisers who continue to make this project possible. And to the AT researchers and clinicians from all over the world, we know how hard they're all working to find life-improving therapies and a cure for AT. And to our family advocates in so many countries, they've been helping us recruit patients to make this as meaningful as possible, to get as much data in here as we can. And last but of course not least, to all the families who are participating and those about to register, we appreciate your logging in and your sharing your data and making such a tremendous difference. This project has been funded in large part by all these families and our grassroots fundraisers at the AT Children's Project, and we've had some input as well from some of the other AT organizations around the world. And we're just truly appreciative of this being such a global effort. And thank you to Sean Kelly and everyone at Action for AT for giving us the opportunity to present today and share what we're doing with the Global AT Family Data Platform. I know we're going to go to questions, but I just want to be sure everyone knows that you can always reach out to me directly at support@atfamilies.org if you have any questions that we don't get to today. And thank you.
That's really good. Jennifer, thank you very much. Questions are coming in, Jennifer, and I think one you can actually probably address straight away because you looked at this. How long approximately does it take to register online, and do you provide updates for those that enroll on what you're doing with the data?
Yes, so we are providing people with hopefully a really simple way to get registered, that it doesn't take long. So from beginning to end, it should take somewhere between 15 and 30 minutes. We don't want people to get too hung up on any particular issues. When you get registered, so if you have any problems with any of the questions or anything else, you can skip it and move to the next, or you can email us and get any help that you'd like. So we're trying to make sure that it's not a cumbersome process. And as for providing information, there have been no publications yet on the data, but we are going to be putting out something in the meantime to at least let people know which researchers are accessing the data and the kinds of questions that they're hoping to answer.
We have another question now coming in from Isabel, and I'm assuming this question is from the perspective of the patient themselves. And the question is it possible to have access to the genetic sequencing of an AT patient who has sent you a sample? So I'm assuming they're asking, as the patient, yes, can you get access to that sequencing?
That's a really great question, and unfortunately, this is considered research grade sequencing as opposed to clinical grade sequencing. And the difference there is there's no one who's actually reviewing the data in order to give it back to a patient, because that has to be, because it's whole genome sequencing, it would be very cost prohibitive in order to do that. So what we do is, as part of the platform, is we get permission that if there's something that we do need to address, that we could get the information to your doctor, who could then order clinical grade sequencing for that particular issue that might pop up. But it is just for the researchers at this point.
Jennifer, can I ask, just in your experience when it comes to collecting data, and this is one of my dumb questions again, how much data? Thank you. How much data do you need to get before you see results? Like, is there a kind of a line that kind of shows, okay, so many hundreds, so many thousands, and then I'm getting a bit more of information, a bit more of a picture?
So I guess it depends on what the data point is. And I'm a social worker by background, so I'm not one of the researchers actually accessing the data and making scientific and clinical decisions. But from what I understand, it depends on how big a difference you'd see, right? So if you see a very small difference between factors, you'd need a lot more patients to participate. If you can see by looking at a piece of data that there are very big differences, then you'd need fewer. But we do know that we have an initial goal of getting 500 patients registered. We feel like that would be a good, robust number to get started. We have 466, as of this morning, 467 people who are registered with their clinical data, and we're closing in on 300 sequences.
Thank you. And just thinking, given how rich and how useful the sort of data that you're collecting on this platform will be, is there any other types of researchers other than those interested in AT who might be interested in the data on this platform?
That's a great question. I mean, we'd be happy to help with findings. We're hoping that data can be accumulated, and that not only that AT data could help others, perhaps including AT data in other larger research projects, that could help AT as well, that it would be reciprocal.
Jennifer, we've had a question from Panos, who's in Greece, but I think this can be applied worldwide, really. Panos says, good afternoon from Greece. Good afternoon, Panos. Says when he'd registered to the platform, he was informed a saliva sample is not possible to be sent to Greece. But I mean, there will be other countries where this can also be relevant. What advice would you offer?
Right. So in the meantime, if you could email us your child's mutation report, if there is one, that would be great. And then what we can do also is once, as we know what numbers of people are interested in different countries, by them getting registered, we've been doing our best to add those countries into our approved countries. And then, we're going to look at other ways too that we might be able to get even more countries involved as time goes on. But it's a very important issue to us. We definitely want to include as many sequences as possible.
Thanks, Jennifer. I think we have one more question from the audience, and this is, would you be interested or would you register and sequence those patients with AT-like disorders who have relevant medical and neurological disorders?
So my understanding, and again, I'm not a doctor, but my understanding is AT, there's something called AT-like disorder, which can be misleading because, of course, the name AT is in the name of it. And so while it shares a lot of symptoms with ataxia-telangiectasia, it is a different gene that causes it. So the information, you can imagine if you had like a DNA-based gene therapy type of treatment for AT, that people with AT-like disorder wouldn't benefit from it if it's something that just addresses the AT gene. So because it's a completely different disease, it would be difficult to include people with ATLD in this platform.
Jennifer, it's been really interesting talking to you. Thank you so much. I know this is at the start of your day, so well done for taking all those questions, and I hope the rest of it is good for you. Thank you again.
Thank you.
Thank you all so much for the opportunity. I really appreciate it.
Take care. We're going to move on to our third presentation now from Professor David Coleman, who is a paediatrician, clinical geneticist and lead of National AT Clinic at Queensland Children's Hospital. Now, you're aware there are time differences from across the world. There's a 10-hour time difference, so we thought we wouldn't wake David up in the middle of the night to do this, so we allowed him to pre-record it.
G'day, Dave Coleman. I'm a paediatrician, geneticist and a metabolic physician here working in Brisbane, and I run the National Clinic for ataxia-telangiectasia in Australia. And we're going to talk today around some novel treatment options that we've developed through some of our research that is also translating into a clinical trial, COVID-19 permitting, that'll be kicking off in the next few months. So we know AT is a very complex disease, and in recent times we've been finding that there are issues with the way that mitochondria work in AT. So mitochondria are like energy packets or the powerhouses of our cells, a bit like batteries for a kid's toy. And there has been some previous evidence of mitochondrial dysfunction, particularly in reactive oxygen species, free radicals, that type of thing, in people with AT. Following on from that, within our clinic and the research we've been doing, we've identified a new role for the ATM gene product in regulating mitochondrial function. This has just been published in Cell Science. And we've basically found that there's a block in how calcium gets transported from what's called the endoplasmic reticulum, which is like a skin on the surface of the mitochondria, into the mitochondria. And calcium's really important for being what's called a cofactor for starting different enzyme reactions. A good way to think of it, the mitochondria are like the engine in your car, and calcium's a bit like the key that goes into the ignition to turn it on. Following from that, we've found that there have been blocks in a particular part of the mitochondria called the Krebs citric acid or cycle, or the TCA cycle, and we think again that's relative to calcium not entering into the mitochondria. In one of my other roles in metabolic medicine is treating people who are born with inborn errors of metabolism. And there's a group of disorders called long-chain fatty acid oxidation disorders, for which we've been using this medication, or its oil triheptanoin, for at least 15 years. And it basically kind of acts as an alternate fuel source which bypasses a block to enter into the TCO, the Krebs citric acid cycle. It's been very useful for some of the inborn errors that I treat, but also for other disorders emerging in the literature, such as epilepsy, a thing called glucose-1 transporter deficiency, amongst other things. Generally very well tolerated, and it's an oil, a bit like MCT oil, medium chain triglyceride, but it's a variant on that. What we've found with our studies is that it rescues our AT cells. So without going through this in a lot of detail, we know that ataxia-telangiectasia cells, either ones that we make in the lab or ones that we take from patients, particularly bronchial epithelial cells, we find that they're very sensitive to glucose stress. So if we introduce a chemical called 2DG into the media that the AT cells are growing with, there's rapid death of those cells. So they're very sensitive to any type of alteration in supply of fuel. When we use triheptanoin or C7 in those cells, they return to almost normal levels of stability compared to normal patients in control samples. And we do see correction of some of the biochemical features associated with TCA metabolism defects as well. Again, a very technical slide, but we found in the AT cells as well, there's not only is there mitochondrial dysfunction that we can measure, but stuff that we can see down the microscope, and that's all rescued when we use C7 with those cells. So what we're finding is that not only are the AT cells very sensitive to nutrient stress, such as not having enough glucose, but we can also rescue them by using an alternate fuel source. What we'll be publishing very soon is that not only does it feed the mitochondria, it actually helps correct the defect and reprogram metabolic pathways. And this was the basis of an application that we put into the Australian Medical Research Futures Fund, and we were able to get $2.5 million out of that to run a clinical trial for patients with AT. At this stage, we're only going to be doing patients in Australia for logistic reasons, but the hope is that we'll be able, if there's an improvement that's measurable, we'll move on to additional families around the world. A key component of this is that our trial steering committee, the people that are helping us run the trial, are very deliberately handpicked colleagues from all of the AT, main AT centres around the world. So it'll be a very rapid way to share information as well if we prove this to be useful. Okay. So with any study, you need to have what's called a primary endpoint, something that's really important that you can measure, and we will be measuring the cell death structures on nasal epithelial cells. Now, realistically, that may not be all that useful for you in the clinic and for you as people, but it gives us something we can measure and have statistical differences on when we supplement people with this medication. More importantly, though, I think we'll be doing lots of other secondary endpoint measures, particularly in and around some of the scales of ataxia. We've been finding with some of our other medical genetic diseases that give you ataxia, we've been identifying that utilising repurposed drugs or medications that have been designed for one thing, but when we use them for another, we have been finding improvement in measurable and reproducible improvements in some of the ataxia-related scales. So I'm quite hopeful that we will see an improvement in and around those clinical areas. And we also have a metabolomic arm to the project where we'll be looking for biomarkers. So something that we can use to say, okay, well, look, this is maybe something we can use to test for not only disease stage, disease progress, but also treatment response as well. Within our local AT team, I don't have everybody involved on that slide. We're very lucky to have a very dedicated group of clinicians and researchers. But the main people running the trial are on the top. There's myself, I'm a clinician. Kate Sinclair, who is our paediatric neurologist who started our AT clinic. Martin Levin, who should need no introduction, is a world expert cell biologist, particularly in the field of ataxia telangiectasia. Ernst Wolvertang, who is a stem cell researcher and has been very useful in helping us develop some of these trials and cells. And Peter Sly and Claire Wainwright are paediatric respiratory physicians, but are very experienced, well-renowned researchers and have been instrumental in helping design the trial. And we've also got some key laboratory staff as well, Abrio in particular. I guess the end game, though, is look, these little cells here are the Purkinje cells, which are the main cells that drive cerebellar function. And they're very, very sensitive to perturbations or changes in calcium metabolism, and very sensitive to changes in mitochondrial function. So I guess realistically what we'd love to do would be to see an improvement not only in how these cells function, but more importantly how that translates into an improvement in quality and quantity of life for people with AT. So we're doing what's called a phase 2A, phase 2B trial, which is where we look at tolerability and safety and efficacy. If it works and we see significant improvements, then we're well placed to move on to utilising a phase 3 trial, which is a multi-site trial. And as I mentioned, our trial steering committee involves people from every main AT research clinic around the world, and we've done that deliberately to help maximise the quality of the trial, but also maximise the dispersing of information should it be successful, because the end result is trying to help people with AT. Look, thank you very much for inviting me to be part of your webinar, and I'm happy to take questions as required. Thank you.
So David is perfectly happy to take questions, but he's in bed asleep. So what we decided to do is ask him our own questions, well, Mark did. Also, we're really aware that loads of you have been bringing questions in through Slido, so thank you very much for that. And what we're going to try to do is get them to David at some point and then get the answers published when David's awake and has time to respond to them. Mark, you spoke to him earlier, and while we were watching that interview, I was trying to get my tongue around triheptanoin.
Triheptanoin, that's right.
Triheptanoin. I'm sure it's a word that flows off the tip of your tongue, but you decided to talk to him, and you had a Q&A session with him, and you were going to ask him about what improvements he expects to see for patients who are actually taking this.
Look, I guess the improvements we would like to see are twofold. Firstly, our primary endpoint measure for the study is looking at cell death from the nasal epithelial cells. We would like to see an improvement in patients in that, which is reflective of what we see in the lab. Realistically, though, that's a means to an end from additional funding to do further studies, because it's easy to measure statistically significant changes in that primary endpoint goal. That being said, what I am most hopeful of is that we will see some improvements in the secondary measures that we're looking at, most notably the ataxia scales, because realistically what we're after is an improvement in function.
Are there any side effects that people might expect to see from taking this drug?
Like any drug, there are potential side effects. I've been using this drug, and metabolic physicians like myself have been using it for at least 15 years for different types of disorders. The side effects are mostly in and around gastrointestinal tolerability, so abdominal pain and diarrhea. It's less likely to happen if we grade up slowly on the dose, and there are also some concerns of it impacting on PEG function, so gastrostomy function. However, there are ways to A, improve tolerability, and B, prevent complications with enteral feeding devices.
Can this be readily purchased over the counter?
Currently, the drug cannot be purchased over the counter. For specified conditions, it can be prescribed through a drug company in the US who got FDA rights to use that for various diseases. For our trial, we are having it manufactured at clinical grade level, but you're not able to buy it over the counter at this juncture.
Assuming the drug proves effective in your study, How long do you think it would be before it becomes available as an approved therapy?
So our current trial will be a 2A/2B, phase 2A/2B trial, which is effectively looking at tolerability and efficacy. If we do find statistically significant changes in AT patients with this trial, the aim then will be to rapidly move to a funded phase 3 international trial. We've deliberately set up the trial with trial steering committee, like an oversight committee, which involves clinicians from each of the main AT clinics around the world. We did that deliberately so A, we could have input onto the validity of the study, but also B, so we could move as quickly as we could, as funding would permit, to do a phase 3 trial such that it would be more available for AT patients around the world.
Do you anticipate seeing a reversal of cerebellar symptoms or just prevention of further deterioration?
I think it'd be hopeful to, and falsely hopeful to, suggest that it would reverse the cerebellar signs. I'd be hopeful to see stability and even some improvement. I guess part of the reason we find that the cerebellar signs progress over time is that the Purkinje cells die, and we can't resuscitate those if they're already gone. But I guess realistically, I'm hoping to see some improvement in scales and stability with luck.
You mentioned that a specific pharma company had gained approval from the FDA for this drug. Does that mean it might be restricted in some way, or would it become generically available to people to use for this indication?
There is a drug company in the US that has distribution rights for this medication for different types of disorders. I have been in contact with them about utilising this in AT should our trial prove successful. As mentioned, we are getting it manufactured at clinical grade via a separate company for this project.
Do you feel that this could impact the time taken to make this available as a drug?
In terms of time frame for being available for the rest of the AT patients around the world, it would very much depend on how we go with this phase 2A/2B study. It's going to run, all the patients will be enrolled over a 14-month period, and then we have 6 to 9 months then of data analysis. As mentioned, we do have a trial steering committee with colleagues from each of the main AT centres around the world involved, such that we would be able to share information. Setting up a trial, though, does take a lot of work. It's taken almost 9 months thus far, to even after funding, to get this to a point where we're ready to start. And that's COVID notwithstanding. So I guess we are hopeful that we will see measurable, reproducible improvements. I'm quite hopeful that we will see some in the ataxia scales, which is a more meaningful functional marker of the disease progress.
Hope you got a lot out of that as well. And thank you. We have noticed your questions are coming in, and we will get them to David as much as quickly as we can and get some answers for you. So, where are we at? We're at the end of the first part of this session of our research presentations. Hope you're enjoying it. I hope you're enjoying the content. And are you surprised, Mark, at how many questions are coming in?
Yeah, surprised and really pleased, actually, that we're getting the engagement from around the world.
And I am the perfect example of a dumb question not being a dumb question. I think. We're going to take a short break now. We'll be back for the final presentations in about 5 or 6 minutes. And during the break, we're going to run our second poll, which we'd love you to take part in. We want to understand your top 3 research priorities. So if you can take part, please do, by answering using the poll button. It's in the top right hand, it will be there, the top right hand corner of your screen. We'll collate the results, and we'll have a look at those after this short break. Go grab yourself a cup of tea or coffee. Welcome back to the second half of the presentations of this Spotlight on AT webinar. You're here with me and Mark. We've got 3 great presentations coming up for you still, but we are going to introduce our next speaker. But first, we asked you to take part in that poll. Thank you very much for those of you who did. And got some interesting results. You're quite surprised, Mark.
Yes, very, very surprised. I think we expected to see that neurodegeneration would be one of the important areas. It's one of the areas that affects people's real day-to-day living. I thought oncology might be higher, though, because of the risk that poses and what people fear long term. But I was also surprised to see that fourth on the list was actually new technologies to improve daily life. That's a bit of a surprise to see that.
Yeah, and emotional and psychological needs, something very, very topical, just 10% at the moment, but obviously that's an ongoing impact for families as well as those who have AT as well.
Yeah, absolutely. Such an important topic.
We'll continue to keep an eye on what you're finding interesting at this moment in time as well. Keep your thoughts coming in and your questions coming in as well, please. And thank you for providing all this useful information. As an organisation which is very passionate about including the patient voice, your feedback is so important to Action for AT. It's going to help guide future funding priorities and ultimately what's important to you as well. Now, let's return to our research presentations. First up, we have Elad Jacoby, and Dr. Jacoby is a paediatric hemato-oncologist and head of cellular therapy at the Sheba Medical Center and Tel Aviv University. So we can say hello. Now, there's a mouthful, isn't there, for a title?
Thank you.
It's an impressive title. Tell us what you're interested in.
So I'm a pediatric hematologist and I deal with blood disorders. And we actually develop and use a lot of therapies that we take advantage of our blood as the immune system to treat cancer and malignancies. And this is what we're going to talk about in this presentation. So I work at the Sheba Medical Center in Israel, which is the largest medical center in our country, and it also hosts the National AT Center in Israel, where we see the patients from the entire country. I, as a hematologist, see them in the case of malignancies, unfortunately. If we go to the next slide. So we know that there's a high incidence of lymphoma, which is cancer of the lymph nodes, or leukemia, cancer of the blood or the bone marrow in patients with AT. And the standard treatment we give to patients, which is high-dose chemotherapy, is very toxic to patients with AT. So this is a great challenge. If we can move on, thank you. So in the past decade, there was a revolution in cancer therapy altogether. And this is cancer immunotherapy. It adds up to surgery and chemotherapy and radiation in treating cancers. Back in 2013, it was designated as the breakthrough of the year in the magazine Science. And in 2018, these gentlemen, Jim Ellison and Tezuka Honjo, were awarded the Nobel Prize of Medicine and Physiology for their achievements in the fields. This was mostly for checkpoint antibodies, which give hope to many patients now, but unfortunately do not work so well for leukemia or lymphoma. So we need even better solutions. So we had to build new immune responses to treat leukemia or lymphoma, which is mostly what I treat. And just to introduce the two major cells that we have in the immune system, one is a B cell that provides antibodies, produces antibodies to attack invaders outside the cell. And the other is the T cell, which is a very powerful killing machine that attacks invaders that go into the cell. Now, to generate a new immune response, we actually combined the two and now made a CAR T cell, which is a T cell that could recognize its target through an antibody. In this way, it's easy to produce and has a very high efficiency in killing tumors. And this is a nice cartoon showing how CAR T cell works. So it's based on a cell that comes from the patient. And then there's a viral vector encoding the CAR, and it has to integrate in the genome and gives the code to design new receptors on the surface of this T cell. And now the T cell knows how to home and attack a malignant cell. In this case, for this example, the CAR recognizes CD19, which is a very common receptor on B-cell leukemia or lymphoma. Once the CAR T cell recognizes the malignant cell, it attacks it, releases cytokines, proliferates, and actually kills the tumor cells. So to start this complex process, we need to take immune cells from the patient, and we take them through an apheresis. It's a process where the blood goes through the machine and just collect the lymphocytes, These are the cells we need to generate from them the good attackers. The T cells there go activation, go processing and transduction with the virus, and proliferation in the lab. After that, they're reinfused to the patient. In the next slide, I just wanted to show two examples of how powerful this treatment is. These are two patients we treated for lymphoma. These did not have AT, but the patient on the left side and the patient in the middle both had lymphoma that was resistant to many therapies before. And you can see this is a PET scan, and most of the small black dots are actually lymph nodes with malignant cells. And you can see that 4 weeks later, after the CAR T cell treatment, these patients were clear from their disease. And this is just by manipulating their immune system. So this is fantastic. So now in 2021, we have already in the market 4 therapies that are approved by the FDA and a lot of other agencies that are CAR T cell based for lymphoma or leukemia. All are targeting CD19, which is limited to B cell malignancies, and all are after the patients have failed 2 lines of therapy before. So these are never given upfront to therapies. So how does all this come to patients with AT? So we know that CAR T cells have excellent results for relapsed leukemia and lymphoma, but in AT, the patients cannot tolerate chemotherapy. So can we use it as first line of therapy? We know that CAR T cells are constructed from the patient's own immune system, but there's an immune deficiency state in AT patients that may limit their effect. We also know that CAR T cells required integration of the CAR virus into the genome of the T cells, and we do not know if this is safe when genome repair is impaired. So this is the basis of what we propose to do in the work. So we're studying potential for CAR T cells for patients from AT. We want to identify if this is feasible, if we can actually generate CAR T cells from patients with AT. We want to study if they are good to clear leukemia in the lab. And we want to see if this is safe, if there's no damage from the viral integration in these cells. I'll show a little bit technical preliminary results we have in the next couple of slides. So we already recruited 8 patients who were willing to donate a small amount of blood for our research. And we also took blood from their parents or sibling, which were heterozygote controls. These had a variety of mutations or deletions and already had 7 products in the lab. This has not been treated anyone, just lab research. We could see, and this is Meir Rosenbaum, he's the PhD in my lab who's heading this work. So this graph shows how well the T cells grow in culture, and you expect them to grow to higher numbers with time. You can see in blue the heterozygote controls grow, but the T cells from patients with AT also grow to less of an extent, but still grow adequately. If we can move to the next slide, please. We also could see that from sometimes very small T cell numbers, you can see in red in the blood, we can get excellent T cell expansion in the CAR T cell product in the lab, both from patients and control. And there's a fairly well CAR transduction. So we could achieve CAR production from these patients. The next stage was to see if these CARs are good. And for this, we tested cytotoxicity. And this is just showing how many of the leukemia cells die in the dish a couple of hours after meeting the CAR T cells. So you can see that about 5 to 10% of the cells die when they meet the CAR T cells, both from the controls and the patients. It's a little bit lower than from healthy volunteers, but we're still repeating these experiments. So overall, we're continuing to test the efficacy of CAR T cells to eliminate leukemia and lymphoma, and we're studying very hard the karyotype and the integration to see if this is a safe therapy. We'll move next to tumor challenge and different CARs. And actually, our vision is that if this is found safe and effective in the lab, CAR-based immunotherapy will be the first-line therapy for patients with AT and malignancies. With that, I'd like to thank many people involved in the work, mostly Mayor Rosenberg and Ifat Zaruk from our AT clinic, Sarah Elitzur, who's heading a lot of cancer data from AT patients, and Gidi Rechavi and Jasmine Jacob, who's studying the viral integration. A lot of thanks for Action for AT for the support, and mostly to the patients and families who are so happily willing to participate in research. I'm happy to take any questions. Back to you, Naga.
Elad, thank you so much, and thank you to your team as well for all their efforts, because so much work goes into this. And Mark, we were talking just while Elad was presenting about the significance of CAR T therapy. Do you just kind of want to go into that area as with your question?
Yeah, so I think what might be useful, Elad, thank you for that, is just to make sure that we really all understand exactly how this works. So really this is around using a kind of genetic engineering, isn't it, which takes the patient's own cell and uses that genetic engineering to create a cell that will recognise the type of cancer. So kind of a cell that you can then put back into that patient. You grow some more, you put them back into that patient, and then it targets that specific cancer and essentially does the body's own job of clearing it away. Is that a reasonable way of putting this?
That's exactly what we do. We would love to give, as little as possible toxic chemotherapy to patients. We love to enhance the immune response that the body can generate. So this is one way to do it.
And does that mean that you have to know exactly what type of cancer it is to be able to do that engineering?
Right. So we need to know what cancer this is, and also we need to identify a target on the cancer cell surface. So something that's around it and just sticks out of it that we can target. And then we can design a CAR, which is kind of a guided missile that can go and target it in the body.
Elad, how long does that process take?
So once you have your CAR designed, it can take between 10 days and about 4 or 5 weeks to get a product from patient's blood back to the patient's blood.
Thank you, Elad. Now, you showed some work that you've already done, which shows that you can actually make these cells from an AT patient, and that it's possible to grow those. So what does this work that you're doing add to what you've already shown?
So the fact that we were able to produce cells did not mean that they work as well as cells from healthy volunteers. We're always concerned that there's certain immune deficiencies in patients with AT that might cause these cells not to work as well. And we have to prove that in the lab. Also, because there's a problem with genomic repair and this process involves genetic engineering, we have to make sure this is safe before we can actually introduce this to patients. These
so,
are
Elad,
yeah.
Sorry, Elad, a few questions coming in. I mean, Isabel's understanding of this is that it's a personalised therapy. And someone's also asking, if you're taking a cell from the patient themselves, does this mean they then have fewer side effects? And great, if so, but also what side effects do people get with this type of treatment?
So if you're taking the cells, it doesn't clearly say that they'll have fewer side effects. If this treatment works, and we never treated this with patients with AT, only with patients with lymphoma or leukemia from other backgrounds. If it works, it may cause vast activation of the immune system. It's called cytokine release syndrome, and it can be like a very bad flu, but it can also require a short ICU admission. So these are significant side effects that we see in patients. Again, this might be different in patients with AT. We still do not know that.
Thank you. And just one more question. Is this then a reactive treatment for patients with cancer, or can it be used as a preventative therapy for people who are at increased disposition?
That's an excellent question. I don't think that it has been used or can be used as a preventive, because you want something preventive to be long-lived, such as a vaccine. And I don't think anyone can use a cell therapy as such at this point. But maybe in the future, if we get good development for that, we can develop some sort of vaccine that would help us in prevention.
Elad, thank you so much. And that excellent question was from Naz. So thank you, Naz, as well for taking part. Elad, thank you. We're going to move on to our penultimate presentation now, and I'm delighted to introduce Ignacio J. Molina, who is a professor of immunology at the University of Granada. Professor, thank you so much for joining us. I've got to ask the obvious question, what is the weather like in Granada? We're not yet into summer.
No, it's getting better, but we had also very winter. It gets very cold in Granada, believe it or not.
Ah, okay. Well, you're making us feel a little bit better here in the UK, at least. I hope all of you are getting better weather than we are. Professor, please, let's hear your presentation.
Okay. Well, first of all, I would like to thank Action for IT and particularly Sean for the kind invitation to share with you what we are doing in the lab, thanks to the financial support of Action for IT, regarding the gene therapy of AT. Before, perhaps it may be a little bit technical, but there is no any other way to go through our project. I want to remind you about the ATM gene. If you look at the far left of the slide, we have the chromosomes where all the genetic information is stored. And in chromosome number 11, in the region that is highlighted in red, we have this gene, which is the responsible for causing the disease. This the ATM gene, and is a very, very large gene, enormous, and that is our main problem. And here you can see how different mutations in different places of the gene give rise to the disease. Now, this belongs to a study that we did a few years ago, and we had identified all those mutations in patients with AT. So the complexity of the gene causes a very complex protein as well, as you can imagine. No? And then you have in the bottom part of the slide the different domains of the protein. Each one is needed for a different function, and also is a very, very big protein. It's very difficult to work with that protein because of its size. And not only that, but also the complexity of the protein. When you look at the three-dimensional structure, which is what you can see right in the bottom center of the slide, so you can see that the protein is folded in a very complicated way. So first of all, what is gene therapy? So gene therapy is we want to define gene therapy as the procedure whereby we are going to insert a gene or part of that gene into a target cell, into the nucleus of a target cell, to modify its properties. And why do we want to do that? Well, one possibility is to modify the function of the target cell. This is very much the approach of the previous presentation. No? For instance, if we have cells involved in anti-tumor response, we can provide these cells with weapons that render these cells particularly effective in clearing this tumor. But in our case, what we want is to introduce an intact gene. Remember that we have patients who have a defective gene. What we want is to introduce a normal gene into the cell of those patients. That's what we call the substitutive or replacement gene therapy. In other words, next, what we are going to do is to insert this circular piece of DNA into the nucleus of a cell that has no function, and with that process, next, we hope to restore the function. That is gene therapy, and this is what we want to achieve, to cure the disease by replacing a defective gene with a healthy gene. But we have a big problem, and the problem is the size of the gene. I have already mentioned that it's enormous, 66 exons, a cDNA of 9.3 kilobases, and that is enormous because if we take as a reference the cDNA of the IL-2 receptor gamma gene that causes an immunodeficiency called X-linked severe combined immunodeficiency and was successfully treated with gene therapy, that gene is only 1.2 kilobases. So compare 1.2 versus 9.3. That is the problem. Next, please. So the traditional approach was to use viral vectors. In other words, what we do is we put the therapeutic gene together with other genes that are needed to produce a artificial virus. And it's a virus that is extremely safe, has been devoid of all areas that may cause a problem into the cell. So it's very safe to use that. And we insert those genes into the cell that is in the bottom left part, which is going to help us in making the virus. So in other words, we put the different plasmids, the different parts of the DNA independently into this packaging cell, and this packaging cell will make the virus for us. And once that the virus has been made, the only thing that we have to do is to put those viral particles in contact with our target cell, in this case, in contact with the cells, the progenitor cells of the patient. And because the entrance process is so effective, we are going to achieve a good efficiency. But again, next, our problem is that the size of the transgene hampers the transduction efficiency. So we are dealing with a problem due to the large size of the gene. Next, please. A few years ago, we started a project also funded by Action for AT, where we wanted to demonstrate whether or not gene therapy was a good procedure for curing the cells of AT. And we took a traditional approach using lentiviral vectors. We put the ATM gene into one of those vectors, and we were able to reconstitute the cells. We were able to force the patient cells to make the products of the ATM gene. So you can see on the right side the white bands, how the transduced cells of the patients show this white band, which is equal to the band that we see in the control cells. And in the bottom, you can see the protein made by those cells, and the wild type cells make that protein that you can see on panel D, and the transduced cells, the reconstituted cells, make a protein of similar size and function. And most importantly, if you look at the bottom panel, when we irradiate the cells with irradiation, we force the cells to repair. That's the key questions, to repair. And we can see that the cells start to make phosphorylated ATM, which is a proof of activation of ATM. Next, please. We went into further detail to demonstrate the function of ATM, and in the wild type cells, in the upper part of the slide, you can see those little green dots that we call foci. that are formed when we irradiate the cells, and those are an indication of the cells actively repairing repairing the DNA. So you can see that very nicely in wild-type cells. You don't see anything in the untransduced cells. And finally, in the bottom part, you can see that when we put the gene into the patient cells, the defects are completely restored. Next, please. We also took other experiments to demonstrate that we could do that in other systems, and here you have more examples. Next, please. Next again. Next. Okay, and the previous. Okay, thank you. And the most important thing is that we have mentioned the sensitivity of patient cells to irradiation. So it was a critical experiment to show the response of patient cells, transduced patient cells, to irradiation, and we demonstrated that patient cells could be restored and became radio resistant, thereby curing one of the most important defects of the disease, the irradiation defects. Next. And also the defects that we see in the cell cycle. Okay, so we were very happy. Next, please. We were very happy because we have demonstrated until that point that gene therapy is able to reconstitute patient cells and to make them to give or to render a normal function. But we ran into a problem that was impossible to solve, and was the efficiency. The efficiency was very, very low. We were not able to transduce more than 20% of the cells, and that is very low when we think in starting clinical trials. Why is that? Obviously, I remind you, because of the enormous size of the gene. So we need alternatives. And this very recently in the form of these new vectors that are called transposons, which are based on mobile DNA elements. That the most important thing is that they integrate into intronic sequences. In other words, these vectors are very safe. When you put these vectors, they go into the DNA in areas where they are not going to perturb the rest of the genome, and the biosafety is therefore very high. But particularly for our problem is that they have a much higher cargo capacity, and because we want to introduce a large gene, we need a large lorry. Just a small van is not enough to carry our high load. So these new vectors show great potential as tools for gene therapy, and there are already 11 ongoing clinical trials. So you can see that they are very innovative, but very promising. So we made the vectors. We were able to make the vectors. We went through all kinds of tests to demonstrate that these vectors were okay. Next, please. And finally, what we have done is to start our experiments, in which we have inserted these vectors into patient cells. So you can see at the bottom left the green fluorescence protein intensity. We put this marker, the GFP, is a very nice way to monitor the efficiency of your transfection, because if you see the green fluorescence, you see also that those cells were successfully transfected with ATM. So the cells on the far left do not show any fluorescence, obviously, because they are not transfected. The cells, when we look at the cells right after the transfection, we saw between and 8 and 15% of transfection, but we were able to enrich those population by a process of purification, and finally, after 2 rounds of enrichment, we have cells that are 95% expressing the ATM cell. So in the moment that we are now, we have demonstrated the capacity of gene therapy to reconstitute the defects of cells from AT patients. But because of the large size of the gene, the procedure using lentiviral vectors is not efficient enough. The alternative came in the form of these transposons, which show a much higher cargo capacity and with equal biosafety. We made 2 transposons containing ATM, and the cells were successfully transfected, obtaining 95% positive cells after enrichment. And we are now working on the analysis of the cells to see whether or not those cells were functionally reconstituted. So we are now working not only in the to demonstrate that the method is safe, but also in the optimization of the transfection method. And finally, next please, I'd like to thank Action for AT for their continuous support. As I mentioned to you, they funded the previous experiment, the previous project, the one using lentiviral vectors, and they have also continued their support to our lab to explore the possibility of moving to this new platform of transposons. So thank you very much for giving me the opportunity to share with you our results. Thank you.
Elad, thank you very much indeed. We have lots of questions coming through. I have one question, and it will tie it into one we've had from Spain as well. With all the research that's going on and you waiting for results, in your head, what's a realistic time frame for you to have some real concrete results that you can actually then put into action in terms of therapy, in terms of, using this gene therapy?
It's very difficult to answer that question because everything regarding ATM gene and its protein goes beyond most rules, no? Because of the gene is so enormous, because of the protein is so complex, we face unexpected problems, no? In other situations, we could go very quick, but considering the difficulties, the inherent difficulties of the ATM gene and protein, I really don't know. It's going to take a while. We are at a very, very preliminary phase, and it's going to be a very, very difficult experiment to do.
Okay, but Ignacio, tell me you're optimistic.
I want to be optimistic, of course, but it's going to take years, yeah. I think that a minimum of 5 years from now.
Okay, Mark, we've had lots of questions coming in, haven't we?
Yeah, unsurprisingly, actually, with this topic, it's a very, very popular topic. We have a question from Cressila, which is, what is the current status of gene therapy with the lentiviral vectors? Is that still in the basic research phase?
No, lentiviral vectors are now the most widely used vectors. They have replaced the traditional oncoretroviral vectors, which were the ones that were used by gene therapy at the beginning. Most protocols now use lentiviral vectors, and of course it was our initial approach, because of the many advantages that lentiviral vectors have. But unfortunately, they cannot handle well enough the enormous size of ATM. But as I said, they have demonstrated already their capability in clinical trials to cure patients, particularly patients with other primary immunodeficiencies.
Ignacio, another question. If gene therapy becomes possible in AT in the future, would it be a one-time treatment, and would you only have to replace the gene once?
Hopefully, we are working with integrative vectors, meaning that we want to target progenitor cells, and I want to say that I'm targeting to cure the immune system problems, okay? And in the hope that by curing the immunodeficiency, I will most likely contribute to diminish the problems of the tumors. But that is not enough for the cerebellar problems, okay? Basically, we are taking one step at a time, and I think that the immune the immune system problems and the tumors could greatly benefit from gene therapy, and we are using integrative vectors, which means that if we restore the hematopoietic precursors with our vectors, the vectors will remain there. So we will not have to give a second dose of the treatment. One will be enough because the vectors are integrative.
Thank you. And as we said, it's a very popular topic. One question which already a few people have also liked and would like to be asked was posed by Molly's parents. And that is, when can patients apply for a trial like this? And I suppose also, how could they apply to take part in a trial such as this?
Well, as I said, we are still developing the vectors. So we are not at the clinical trial yet, okay? We are not even the clinical or starting the clinical trial will be hopefully the endpoint of our project, because everything that we are doing is in the lab right now. We need to have the tools. We need to have the elements able to carry the gene into the patient cells in an efficient way. And this is what we are working on right now, because we don't have such a tool. We don't have the elements needed to introduce the cells efficiently into patient cells. As soon as we finish that, if we are successful, and if the preclinical data are convincing enough, we hope to apply for a clinical trial, because obviously that is our ultimate goal.
Thank you. I think one final question, and this one's an interesting one. How much will safety need to be demonstrated when you get to the clinical trial and then onwards into a treatment? How much will safety need to be demonstrated to the regulatory authorities before an AT child can actually be dosed?
Okay, we learn a lot from the problems that arose in other immunodeficiencies. Okay, so right now we know very much what to do, and basically want to demonstrate is the random integration of the vector, and it's something that can be done easily. It's not a big problem. And again, in terms of safety, we have benefited enormously from the previous experience. For instance, the X-linked severe combined immunodeficiency is the disease that was the first to be treated, and it was the first to show us the areas where we had to improve the biosafety. So now we know what to look at, and that is a tremendous advantage.
Ignacio J. Molina, Professor of Immunology at the University of Granada, thank you for your hard work. I'm going to be optimistic that it's quicker than 5 years. I'm going to bring that optimism for you, and that you'll get some sunshine too.
Thank you very much. Thank
Do
you.
take care. Thank you. Thank you also for all of your questions that have been flooding in. It just shows how engaged you are with this and with the research that's being presented. We're going to move on to our last, but by no means least, presentation for the day. Dr. Andrew Prale is the presenter, and he is a locum consultant in paediatric respiratory medicine at Nottingham Children's Hospital. Hello, a smiley face there in your very neat office, may I say. Please let us know your thoughts.
Thank you. Well, I am delighted. I have to confess, the view of the office is of my office colleague, and his desk is tidier than mine. So thank you for the opportunity to talk today. And I'd really just like to share some work that we've been doing funded by Action for AT, and why we're doing it, what we hope to get from it, and we're partway through the project, and what we want to do next. So what work are we carrying out? So if I could have the next slide, please. So this is Nottingham. This is just outside my window. This is not today, but this is one of the few sunny days of the year in Nottingham. And this is Queen's Medical Centre. For those of you from the UK who may be watching, you'll be familiar with coming to Nottingham. Hopefully, patients and parents would be, because you'd come to Nottingham for the annual clinic, and that's actually at the other hospital. And we're doing our work here at the Sir Peter Mansfield Imaging Centre, which is where I am at the moment. And this is a very child-friendly advice. So I'm a paediatric respiratory consultant, or in the United States and elsewhere in the world, we're often called paediatric pulmonologists. And I do my research with the University of Nottingham here at the Sir Peter Mansfield Imaging Centre. This is a picture of me having one of the MRI scans, and I'm interested in thinking about how we can improve the treatment for children with AT, and particularly improve the lung health of children with AT. And so this is me having one of these lung scans, and this is one of my colleagues, Brett, and he's just putting me inside the scanner. I think it's important that I know what I'm putting children through when we do research for children and on children. And so I've had all the tests that we do here. We have this amazing facility which Rob Dineen, Professor Dineen, showed earlier, which allows children to come for a reasonably large period of time and be in a relaxed environment, where children can settle and have a bit of time whilst we talk through the research with their parents and guardians, and then after that we do some lung scans. So I really wanted to talk about why we do these lung scans. Oh, and I should mention that we can show people with this amazing Lego model of our MRI scanner exactly what a lung scan would show to a child, and you know what it would involve, and where their parents would be, and so on, and that's very reassuring to children who are taking part in our research. So what are we trying to do? So what we're trying to do is improve treatment for the lungs. And there's a reason for that, and that's unfortunately children, young people, adults with AT can run into lung problems, and we really want to be in the business of reducing the severity of these and stopping them from having an impact on people's lives. And our challenge is measurement of how healthy the lungs are, and that's our current main problem. So many of you will be familiar with going to see a doctor, and you may also be familiar with having lung tests done. And typically these would involve blowing into devices. So this is a graph of children and young people with ataxia-telangiectasia. And on the left-hand side, that scale from 0 to 100 is measuring the lung function through one measure. And this is how much air they can blow out of their lungs in one big, huge breath. And as you can see, we've done this on children from the age of 5 all the way up to just past 18. Each one of these circles is one child, on one measurement on one child. And then if we've got more than one measurement per child, we've we filled in a line between them in grey. And this allows us in clinic to track how people's lung function changes over time. If we could go to the next slide. So what I want to show you here is this is one of our patients in clinic, and you can see that their lung function is very stable over time. And we look at this and we give some confidence to saying, well, actually, this patient's lungs look okay, at least by this measure. But then if we go to the next slide, this is a different patient, and you can see the first time they had a go, they scored roughly 30%, and then the second time, only a few weeks later, they scored over 60%, and we hadn't done a lot to change their lung function in that time. And so this isn't that this particular patient isn't trying hard enough with their lung function, far from it, but what it is the technique of being able to blow hard and long and do things like lung function tests is very difficult if you've got ataxia. And what that means is that despite people's best efforts, we find lung function that is very low and very high, and it's very difficult to interpret it. So if we go to the next slide, what this means is two things. One thing in clinical practice, though we do these lung function tests, we always think about symptoms and how people are describing cough and other things in order to interpret the lung function tests themselves. And the second thing is that ultimately we want to be able to try new treatments for the lungs in AT. And there's other diseases where there's been a lot of success in improving treatments. And an obvious example is something called cystic fibrosis. And those trials needed hundreds of people in those trials of new medications in order to demonstrate that these treatments worked. And if you have a test of lung function which works worse in children and young people with ataxia telangiectasia, that means that you would need even more people in trials. And so what we're trying to say is that our current best tests that we use in clinic really aren't good enough either for clinical practice or indeed to study new medications. And so I will be in a position to say to you in a few years' time, we should try this treatment. It works in ataxia telangiectasia. So if we have the next slide, please. So essentially a better test would mean that we wouldn't need as many patients in trials, and a better test would also mean that we would be able to know for individual patients when we see you in clinic how well your lungs are doing and do we need to prescribe any new treatments. Next slide, please. So what we've been doing is we've been looking into a particular type of MRI scanner. So for those of you who are here right at the start of this afternoon's amazing session, Rob Dineen was talking about magnetic resonance imaging, which is a type of scanner. And as he was pointing out, it doesn't involve any radiation, which of course is a significant concern for people who've got ataxia telangiectasia. It allows you to do these scans with normal breathing, this particular technique. And we can look at both lung structure to a degree, but also lung function. So this patient here is not a patient with ataxia telangiectasia. This is on the top a healthy volunteer, and on the bottom a patient with a different type of lung disease. And where the air from the atmosphere is getting into the lungs, it shows up very bright with yellow, green, and then a bit less air in blue. And then you can see on the bottom, this is a patient with a different lung disease, and the lungs aren't working very well, and you can tell that because there are lots of areas of the lungs which are not very bright, and there are areas of the lungs where you should see a bright colour and you just don't. So if we go to the next slide. So what we've been doing in this study is we've been looking at a group of children and young people with ataxia telangiectasia, and we've measured their lung health using this type of MRI scan. And we've completed the baseline, which is the first on the left study here. And then what we plan to do is follow up individuals who've been in this study and do repeated scans, and that will allow us to work out whether people with ataxia telangiectasia can have repeated goes in the scanner and will cope with that, and also what we see about how variable this test is to allow us to say, well, if we do this for you in clinic, how reliable is it? This is an example from one of our studies. We get a variety of different information from each scan. So how the scan works is that you lie in the scanner and you breathe normally, and you have a loosely fitting face mask over your face, and you breathe in oxygen. And as you breathe in oxygen, what the scan measures is where that oxygen gets to in your lungs. So on the top level here, we've got different slices through the same person's chest, and in the very bright sort of yellow and green, that's where a lot of oxygen is getting to, and where it's darker and blue, you've got less oxygen getting to. We can also measure how long it takes oxygen to get into your lungs. And we can also, which is the second line, as you can see the bits there that are very bright yellow, and those are areas where it takes a long time for oxygen to get into the lungs. And then on the bottom, we've got delta R1, and what that means is how high the oxygen level gets into your lungs when you are given oxygen. And this is reasonably helpful because it tells us which bits of the lungs quickly get a large amount of oxygen. Next slide, please. So we can zoom in on a particular image, and you can see here on the right-hand side, that lung, which is actually the left lung, because this is somebody looking straight at it, has an area where there's not a lot of oxygen getting to, and that's an area of interest that we would like to follow up and see what it looks like on the next scan. Is this something as a one-off on that day, or is this something that's always there for this particular patient? Next slide, please. The other thing that we can do is we can zoom in on lungs, and we can think about the whole three-dimensional structure of the lungs. So oftentimes when we look at scans, we think about just a two-dimensional image on X-rays, but with these scans, we can look at the whole three-dimensional structure of the lungs, and we can work out which lobes of the lungs are working well and which lobes of the lungs are not working well. And this is just an animation to show how we can do that for participants who are in our study. So we've been doing this work, and we've done our first group of participants, and that's worked really well. And we got some really great feedback from people who took part in the study about what they enjoyed about the study day and how they'd like us to adjust our scans and make them very child-friendly. So what we've been doing, in fact, during the lockdown period and for a period just before is improving the scans ever so slightly, but also thinking about how we can make it more comfortable for participants. So we've now got a new scanning system which has music in, so we're using a different scanner for that. And we're also thinking about using a scanner which is slightly less claustrophobic. I've been amazed at how well children with AT can cope with us doing things, but we'd like to minimize the distress that involves, and so we've shortened the scan time to 7 minutes, and we're now using a scanner which is wider, so it doesn't feel like you're so closed in. So we've done our first lot of scans, and we are going to contact some new people to take part, and also go back to people who've had one scan and ask them if they'd be happy to come for a second. And then we've also got to look to the future about thinking about how young we could go with our scans. So currently the youngest children who've been in our scanner, because we want to catch lung disease early on, is 7 years of age. And we haven't managed to get anybody younger than that to go through this scanning sequence. But we would like to be able to look at very young children. So this is our next scanner that we're going to be using. So this doesn't give us as detailed information about the lungs of children, but our hope is in the future to use this particular scanner where we've had 3-year-olds in it to measure lung function with. And this works very nicely in that the young children can sit on their parents' lap or somebody who they trust's lap, and they can have a scan and they can watch television at the same time, and they cope with this very nicely. So the ultimate aim is to be able to work out what treatments to use, both in clinic to be able to say, well, actually your lungs are slightly worse than they were last year, so we'd like you to do more physiotherapy or so on. And then the second thing is when we get treatments for AT, we would like to be able to make sure they help the lungs. But also there are many treatments that we currently prescribe in other clinical settings, in other respiratory clinics, that potentially would be useful in AT. And if we have a very nice method of measuring lung function, then we would be able to hopefully fairly quickly do clinical trials of these types of treatments. So that's really all I had to say. I really have to thank Action for AT. None of this work would happen without Action for AT support. None of it would also happen without parents and young people being willing to come to Nottingham to have these scans. So thank you all. Thank you.
And Andrew, thank you very much as well. And before we get to the serious questions, have you thought about a decent playlist for the children? Because, that's how you connect with music.
Well, we haven't actually, but we'd be very happy to take some advice on what would be a good playlist to have.
Suggestions will come in. Mark, do you want to go to the serious questions? We've had a lot come in.
Yeah, again, quite a few questions on this topic. Thank you. And thank you, Andrew, for such a clear presentation. So one of the first questions is really around what do we do with the results of this MRI scan? How do you transfer the results of the scan that you see into a decision around therapies or treatments?
That's a really great question. And so when we designed the study, what we wanted to know is if we see something on one scan, are we going to see it on the next scan and the next scan? And if we introduce a new treatment, will we then say, oh, well, this treatment is working very nicely. We'll see an improvement in the scan. And that's why we really want to take the same group of children and scan them on several occasions, because that would allow us to say, well, actually, a one-off change doesn't happen in every single scan, but if we actually find a change on the first scan, is there in the second, there in the third, then that will be very useful. So what these scans give us at the end of the day is a three-dimensional picture of somebody's lungs, and also a number about how well the lungs are working. And then what we can do is, you can imagine if I saw you in clinic, that I would say, well, this is what the number is, and this is where the problem is in the lungs. Let's prescribe this treatment and then potentially repeat the scan next year and see if it's improved. And that, I think, is our hope, that we would be able to interpret them and tailor the treatments for individual children and young people with AT.
I'm very conscious that we're coming up to our 2 hours, but we're going to make sure we just get a couple more questions in with Andrew. Andrew, are AT lung problems a result of lung cells lacking the AT gene, independent of the cerebellar atrophy? I've just lost that question, but that's the first part. That's the first part of that question.
That's such a great question, and I would love to be able to give you the answer to it, and I'd love for anybody in the world to be able to give you the answer, but I think what we can say is, at the moment, we don't know how much of lung disease in AT is due to lack of ATM, the protein that causes ataxic telangiectasia, being functional in the lung, how much of it being due to not being able to swallow particularly well, or having reflux of stomach contents and getting up into the throat and then back down into the lung, or indeed the difficulties with the immune system, or indeed the difficulties when you've not got great cerebellar function in being able to do things like cough effectively. And I am a pragmatic person, and I think that probably it's a combination of all of these factors which lead to people with AT running into lung problems. I think that's all we can say at the moment.
Well, you answered the second part without even seeing it. I just thought I'd let you know, because the second part was, or does reduced muscle control contribute? So there you go, you've answered it anyway. Mark, we've got one more.
Yeah, one more question, thank you. And the question is, if you develop a test based on this MRI approach, will it allow you to prescribe medication that hasn't been used in AT before, but is perhaps used in other conditions, to see whether it would help?
That's a great question. So I think there are some medications which are used in other diseases that clearly would not be useful for AT. So there are those medications that are tailored to specific diseases, that are specific gene therapies for other diseases, that we would not be able to use because they just wouldn't work. There are other medications that we perhaps use more aggressively in other diseases. For instance, aggressive use of antibiotics, aggressive use of nebulized antibiotics, differences in approaches to physiotherapy. All of these things could be quite nicely moved from one respiratory clinic into the AT clinic that would potentially be available. And right now, I have reluctance in prescribing some of them because I don't know what they will do to children with AT's lungs. So I think there are medications that we could try almost immediately. And then, of course, there are medications in the future that we hope are being developed now, or we know are being developed now, that we hope will be available to us. And these tests hopefully will tell us, well, do they help for the lungs, which, of course, can be a severe problem in AT.
Andrew, I'm glad you're sitting down. I'm not sure how easily you blush, but let me just tell you a couple of comments that are coming through. Does Dr. Prowl promise to focus on AT forever? He's wonderful. And we don't have many lung experts who know AT and keep up with the new technologies. And someone else has added to this comment, can I agree that he's wonderful? As are all these dedicated science and doctors. Thank you to them. So look, you can't continue forever. I don't think science is quite there yet, but promise everyone here that you're not leaving just yet.
Oh no, I mean, people who come to the Nottingham Clinic know that I'm often there and I have no plans to stop, so thank you.
Oh, look, you see, you are blushing a little bit. I hate to embarrass you, but you are blushing a little. We made you laugh as well. Listen, Dr. Andrew Prowl, thank you so much. It's been a marvellous way to round up our presentations, and your enthusiasm is, honestly, it's infectious, but also provides a lot of hope as well, and a lot of security for all those families, I think, who are watching and who are keen for the research to progress. So thank you for your work, and thank you to your colleague for the very neat side of the office as well.
Thank you.
Thank you. It's what it's all about, Mark, isn't it? In terms of passionate, in-depth, hard-working scientists that you've got on the team, that you've brought on board for this as well. It means so much, and the work's going to continue. Yeah, I mean, exactly. The research is being conducted by people who are passionate, and is being funded by people who are passionate to fund that research. So it's just fantastic to have the opportunity to hear from all of our presenters today, and Andrew as well, and just hear their passion and the work that they're doing. And I will just echo, someone said, thank you, Naga and Dr. Toms, thank you for engaging with us today. And Naz has said, who asked a question earlier, he said, as a parent of a child with AT, I can't thank, and Mark, you'll appreciate this, I can't thank Action for AT and the presenters enough for organising it. It's going on nearly 2 years since seeing a specialist, and I can only say that to all of you who are experiencing this and who are invested in this, thank you for your support as well to Action for AT. I mean, that support is invaluable, isn't it? It's just so important. Without us coming together as a community and without the support that we get, we wouldn't be able to fund this research and ultimately do what we're trying to do, which is find treatments or a cure. I'll do the official stuff now. We've said thank you. Thank you so much for being with us. I hope you have a greater understanding of the highlighted projects. I certainly do. Recordings of all the presentations are going to be available over the next few days, and Action for AT is going to be in contact with all of you to provide download links and request your feedback about today's event. In the meantime, of course, you can visit the Action for AT website to see details of all the studies that they're currently funding, or if you want to donate, if you can donate, so that they continue their investment into the wonderful work you've been getting some insight to as well. You can click on the 3 lines in the top left-hand corner of Slido and you'll see a link which says donate to Action for AT. And before we leave you, I'd like to thank all the speakers and, of course, Dr. Mark Toms for being the brilliant brain here who's explained loads of stuff to me all afternoon and engaged with the questions as well. Thank you. I know this has been really important to you, and everyone's taken so much time out of their busy schedules, and they are busy because they're doing brilliant work, and let's hope they continue to do so, and I wish them every success. Hope you found this informative. You can use the poll button one more time to provide your overall feedback. Dr. Toms, I'll give you the chance to say goodbye. Thank you very much and goodbye. Take care.